This is the Introduction to Anatomy and Physiology 3, which is the nervous system. We're going to talk about the muscular system and the reproductive system later. For today's purpose, we want to cover 6 objectives that are listed on the board. I'm going to start with Objective 1. Objective 1 asks you to differentiate between the central nervous system and peripheral nerves, and also asks you to identify the fact that electrical impulses are carried by nerve cells. So, in this image, you're going to see the central nervous system in green and the peripheral nervous system in pink. Central nervous system is composed of the brain and the spinal cord. The peripheral nerves are all other nerves that go out to the cells of your body. Now, this is going a bit beyond what the objective is requiring of you, but I thought it was interesting for us to understand. So the nervous system can be divided into the 2 parts we just discussed: brain and spinal cord, the central nervous system, and the peripheral. The peripheral nervous system is then subdivided into 2 categories: sensory neurons and motor neurons. And they actually make sense, right? Those headings make sense. Sensory neurons take in information from your environment. They sense information in your environment. Motor neurons are going to carry out actions in your body. And motor neurons have 2 subcategories, which are somatic and autonomic. And I remember this: autonomic is like automatic. It happens involuntarily, whereas somatic happens, those are voluntary functions. So I see a bear, I run away, that's going to be somatic. I swallow and peristalsis occurs, that's autonomic. Right? Anything that is occurring without voluntary action is autonomic. So swallowing, we also know that when we're breathing, you're not thinking, contract the diaphragm, relax the diaphragm, contract the diaphragm, relax the diaphragm. It's an involuntary process and it's autonomic. Now the autonomic system can be subdivided into 2 categories: the sympathetic and the parasympathetic. Sympathetic is known for fight or flight. And it's about stimulating the body in emergency situations. Okay, this is a lot of text, right? And this is beyond what you need to know for the objective. Just thought it would be worth our while to take a look at it. Questions so far for me? Yeah?
What was the other one? The sympathetic?
So yeah, this is the sympathetic, and you guys can read this more on the Prezi on your own time. The other one is the parasympathetic, which is commonly referred to as rest and digest.
Is this part of the objective?
Nope. The objective, all you need to know is the central nervous system is the brain and the spinal cord. Peripheral nerves go everywhere else. Yes?
If you were to reference it, would you say parasympathetic and not rest and digest?
Yeah. If you really like this information, what I just showed you about the divisions of the nervous system, and this is something you want to study in more detail, select option E in the spring when I let you choose options and dig really deep. That's neurobiology and behavior. It's a cool option. And we're just scratching the surface today about the nervous system and nerves in general. So if you really like this, dig deep by choosing option E. Okay? All right, now let's go back to the objectives. We're good on Objective 1. Let's look at Objective 2. Questions about Objective 1 first? No questions about Objective 1?
Oh. No, I was just confused about why I was looking at nerves.
Oh yeah, because there's 3 sections in 11. Yeah. All right, so let's look at Objective 2. Draw and label a diagram of a motor neuron. So again, I'm going to give you a little bit more information in this diagram than necessary. I'm going to tell you the 4 things you need to know. If you're interested, if you're someone that's like, hey, Stickgraph, just tell me what I need to know for this course. You only need to know 4 things about a motor neuron. And that is the cell body, which contains dendrites. These dendrites are extensions, and that's not actually on here, it should be. Dendrites. Which receive a signal and pass it along the axon. That's the second thing you need to know. The axon can vary in length. Some axons are extremely long. They look like pieces of spaghetti. Anybody go to the Human Bodies exhibit when it was here? Yeah, did you guys see the nervous system when they dissected everything away and all that was there were the nerves? You remember seeing some really long nerve cells? Those are axons. So the axon carries the signal down the length of the nerve to the axon terminal. That's the third thing you need to know. So this is where the signal is going to leave this nerve cell and pass to the next one, the axon terminal. And the axon is actually protected by an insulative layer called a myelin sheath. The myelin sheath is composed of proteins and lipids, and it's like the rubber that we put around our electrical wires, right? So birds can sit on electrical wires because they're insulated by rubber. The myelin sheath insulates this axon so that the electrical signal that gets passed doesn't actually end up affecting other cells. The purple is the nerve cell, okay? And the purple actually travels through these Schwann cells. So the yellow are different cells. The purple is all one big cell. And the yellow cells are smaller cells that insulate and wrap around the purple cell. Are we clear on that? You do not need to know Schwann cell for this course. That's additional information. There's also another piece of this diagram that we're going to talk about, and that is not required for the course, but it's helpful information. That's going to be nodes of Ranvier. Ranvier was the French scientist that discovered that there are actual nodes here between cells of the myelin sheath. We'll talk more about those in a little bit. So 4 things you need to know on this diagram for this course. Dendrites receive the signal. That's these. The axon transmits the signal down the length of the nerve. It's insulated by what's called a myelin sheath, and it ends at the nerve cell at what's called an axon terminal. Questions about those 4 parts of the nerve cell? Moving on, you're going to see that it's actually a connection. The nervous system is a connection of nerve cells to each other. So you're going to pass a signal from the axon terminal of one nerve cell to the dendrites of another nerve cell. So these are connected to each other. The axon terminals pass the signal to the dendrites. So those are the first 2 objectives. Questions about the first 2? Let's talk about the third objective. Sensory neurons, relay neurons, and motor neurons, and how these operate together. So, when a stimulus is received by your body, sensory nerves pass the signal from the receptors to the central nervous system, where the relay neurons transmit it to motor neurons that cause an effect in the body. Right? They result in some sort of change in the body. So, yeah, sensory neurons take in sensory input from the environment, stimuli from the environment. That's the role of sensory neurons. Relay neurons exist within the central nervous system. So sensory neurons bring information from your senses, from your body, to the central nervous system. Those are peripheral nerves. So sensory nerves are going to be peripheral. We then hit relay nerves, which are part of the central nervous system, that are then going to pass a signal via motor neurons to actually change something about the body. And I'm going to give you an example of this. In this pathway, the circles represent the cell bodies. The long string represents the axon. And the V structure, I kind of feel like it's a little misleading to you guys. Those are not arrows. Those V's are not arrows pointing. They're actually axon terminals. So the signal passes from the receptor in the skin through a sensory neuron with this long axon down to the terminal, the axon terminal, where it's transmitted from the sensory neuron to the relay neuron. This big gray substance represents a vertebrae. So in your spinal column you have discs, you have vertebrae. Anybody heard of a herniated disc before? Yeah? So what can happen there is that the disc can actually slide out of place, which is going to be in between the vertebrae and can put pressure on nerves. So that can obviously result in some pain for the individual with a herniated disc. There are treatment options for that, which we can talk about another time. It's kind of aside from this. Anybody had a herniated disc personally? No? Okay. All right. So at some point remind me to talk to you about it. Okay. So we've got the sensory neuron transmitted the signal to the vertebral column, the spinal cord, via relay neurons, so it's received at relay neurons. And if it passes the signal directly back via a motor neuron, it causes contraction of skeletal muscle. That's what's known as a reflex arc. So this image here shows a reflex arc. Why are reflexes important? Yeah.
Are reflexes autonomic?
Yeah. So why would the reflex be important? Let's say I put my hand down on a hot surface and I don't have any reflex. Yeah.
They keep you from hurting yourself?
Yeah, reflexes are going to prevent you from injuring yourself. So if I put my hand on a stove that's hot, without even realizing what I've done, I will draw away from that immediately. Because it bypasses, although it transmits to the central nervous system, it doesn't go to the integration system in your brain. So relay neurons can actually transmit the signal to your brain for integration in your body. So when you're learning something, like hopefully you are now, or you're thinking about something, or you're memorizing stuff, or you're realizing emotions that you have, those are all being integrated in your brain. Whereas reflexes travel through an arc pathway that go directly back to the system that detected the change in the body. So questions about that? Let me go back to the objective. I don't take questions about it. Yeah.
So this is exclusively not related to the brain, so this is just like functions that happen almost automatically.
A reflex arc is not integrated in the brain. It's not your body saying, my hand is on a hot stove right now, I should probably move it, right? It happens instantly or near instantly. I'm going to talk to you about the milliseconds that it takes for nerves to pass signals. So let's take a look at item number 3. We understand that we get receptors that have sensory neurons, then goes to relay neurons in the central nervous system, and then to effector via motor neurons. Yes? Questions about that? Number 4, resting and action potential via depolarization and repolarization. So I've got 3 images to help us understand this one. This is the most basic image, and it defines the phases. There's 4 phases that nerve cells go through. So this is a graph that shows time on the x-axis and membrane potential. So this is measured in millivolts. So this is an electrochemical gradient that's actually present in your nerves right now. Nerves that are at rest are chilling at about -70 millivolts. That's phase 1, a resting nerve. Inside the nerve, the measurement of the electrochemical gradient is chilling at about -7. When it receives a signal that causes action, it's going to enter the second phase, which is the depolarization phase, in which the nerve cell goes from being -70 all the way up to +50 millivolts. At the top, this is called the action potential. So once it reaches +50, it goes into the third phase called repolarizing, where it's going to, again, go back down to the negative, except it actually goes beyond -70. It goes down to around -90 millivolts. That's called the undershoot. Any idea why a nerve cell, why it would be beneficial to have an undershoot?
You don't fire it off?
Yes. So it actually prevents overstimulation of the nerve cell. If it just went resting, depolarizing, repolarizing, resting, it could continually be stimulated, and that can result in overstimulation. So the undershoot prevents it from being overstimulated, right? If you think about this, your body actually has ways of recognizing common sounds, right? You've got this vent blowing right here, which you guys hear every day, and you probably just realize it's background noise. Do you think about the fact that you're wearing clothing? Do you have nerves that are transmitting signals by, oh my gosh, I got a shirt on? Oh, no, right?
No.
Okay, so your body has to process so much sensory input that it's going to rely on its nervous system to identify pertinent information from that which is irrelevant. Clobro, you had a question for me? No? Okay. So this entire cycle is referred to as an action potential. It also refers to the peak value there. So there's an interesting period called the threshold. If a signal does not go beyond -55, it's a failed initiation. So the nerve receives a signal, but it never passes that signal down the axon to the next nerve cell because it hasn't reached what's called the threshold potential. The action potential is an all or nothing event. If it fires, if it gets past the threshold, it's going to transmit that signal down the nerve to the next nerve in the sequence, or to the effector cell. It's not a matter of how hard the signal hits the nerve, it's how frequently the signal hits the nerve. Frequency is what determines the magnitude of that. Yeah? Question? Fish sticks?
Nothing.
No? So these represent failed initiations. They don't pass the threshold. Once threshold is passed, it's going to complete the cycle. And this graph gives you a little bit more information about time. To go from a resting nerve, passing the signal down to the next nerve and going back to rest takes about 4 milliseconds. So a millisecond is 1/1000th of a second. Faster than any computer ever known to man. Your nerve cells can transmit electrochemical signals faster than any computer ever known to man. 4 milliseconds to go from rest to rest. Complete the cycle.
Is it possible for something to happen where it slows down, like it causes it to slow down?
So that's a good question. Is it possible for this process to go slower or faster? The diameter of the axon actually can make a difference. But we're talking very specific points about this. That, remember, this is just our general overview. So if you want to study neurobiology in option E, you can learn more about that. Yeah?
Because this cycle is receiving the electrochemical signal
Yep.
and releasing it, or
Yep.
just released?
No, it's receiving, releasing. So this is from a resting nerve cell getting the signal to the dendrite, passing it by its axon through the axon terminal. Yes?
So then what does it look like? So if there's a failed initiation,
Yeah.
do you just not get that because you're...
There would be no reaction there. So the nerve's not stimulated to cause the signal to pass.
Okay.
All right, let's go to the next one, which is a little more colorful. Talks about the phases and what's happening with membrane proteins. So picture the axon of a nerve cell. That axon has a membrane that's going to allow the passage of substances from the environment into the nerve cell and out of the nerve cell. So at rest, at -70 millivolts, in the initial millisecond here, both sodium and potassium channels are closed. Now let's talk about sodium and potassium for a second. On the back board there's a periodic table. In what group would you find sodium and potassium on the periodic table? Group, periodic table, right? Groups referring to columns. So which column? The first one. The first one.
Yeah.
So what would the charge of a sodium ion and a potassium ion be? One. One negative, one positive, which one?
Negative.
Nope. Try again. One positive. So sodium has a positive charge, potassium has a 1+ charge. You guys are with me in that, yes? Now, here's what's going to happen. Your body establishes concentration gradients and allows the diffusion of these ions into and out of a nerve cell to change the membrane potential. So it starts out negative. Both channels are closed, sodium and potassium. Once threshold has been reached, sodium channels open and they stimulate more, this section here that says more sodium channels open, those are called voltage-gated ion channels. So because of a change in voltage here in the yellow phase, where sodium channels open, it stimulates other voltage-gated ion channels to open up and allows sodium to flood into the cell, the nerve cell. So we're bringing a lot of sodium in. Sodium has what charge?
1.
+1, right? So if we're bringing in a lot of sodium, we're going to actually extend up into the positive range. That's called depolarization. After we hit our maximum positive, the sodium channels close and potassium channels open up. Potassium is in high concentration within the nerve cell And I've got some animations to show you tomorrow about this that'll put it in action for you. Potassium is in high concentration within the nerve cell and low concentration outside of the nerve cell. So if you shut off the valve allowing sodium to come in and you open up the valve allowing potassium to go out, you're removing positive charge from the inside of the nerve cell. By removing that positive charge, you're actually making that nerve cell more and more negative. That's repolarization. The reason there's an undershoot is that the potassium channels do not close. There's a delay in their closing. They do not close until time has gone by. So this blue region on the graph represents the time that the potassium channels are open. They're open from the top until we end our undershoot. So what would reestablish the concentration gradient? Sodium was in high concentration outside, we allowed it to flow in. Potassium was in high concentration inside, we allowed it to flow out. Any idea what type of protein would reestablish those concentration gradients? From something we learned last year, the sodium, potassium, yes, you said it. Yes, sodium potassium pump. The sodium potassium pump moves 3 sodium ions out of the nerve cell and brings 2 potassium ions in. I've got images and animations to show you all this, okay? So, 4 phases you need to be aware of in what's called an action potential. The resting state, depolarization after the threshold has been reached. Repolarization, reestablishing that negative value, actually travels below -70 in what's called the undershoot. And then the sodium potassium pump to reestablish those concentration gradients at the resting state. We know the undershoot is important to prevent overstimulation of the nerve. Questions about objective, what is this, 4 or 5? Where are we? 4? Yeah.
So you said that computers have a slower response time than nerves.
Yeah.
How is that?
I don't
Just
know.
like computer to computer interaction still is faster than a nerve. What makes the brain faster is the parallel processing.
So, good. His question is, I said earlier that the human brain functions faster than any computer, right? And he's saying, well, you can get computers connected to each other, transmitting information faster. But the amount of information
Yeah.
that your body is subjected to, that the brain has to actually integrate, is greater than that when you connect 2 computers to each other.
Yeah, so it's the parallel processing power, like the ability to process multiple things at once that makes it faster.
Yeah.
Okay.
Good. Good question. Other questions about the first 4 objectives? 2 left. Yeah.
There are more objectives.
Yeah, I can get you more. Let's look at number 5. How does a nerve impulse pass along a non-myelinated neuron? So there's 2 types of neurons that we're going to look at. The top one is a myelinated neuron. It's got a myelin sheath, and I told you that we would talk about nodes of Ranvier, right? Ranvier is that French scientist that discovered this. In a myelinated sheath, there are actually regions between the Schwann cells that have high concentrations of integral proteins that allow ions to flow into and out of the nerve. So in a myelinated sheath, you've got these regions where you have ions that undergo rapid exchange. In a non-myelinated neuron, you lack that myelin sheath, you lack the Schwann cells, and so you end up with a continual processing of the action potential. So the action potential begins here in the axon and travels in one direction down the length of the axon to the next cell. What prevents it from going backwards? It's all about the membrane proteins that are allowing the passage of ions into and out of the cell and the establishment of concentration gradients. Tomorrow, a couple of animations will make this more clear, that actually show you the transmission of a nerve signal. Okay, this diagram was actually in your textbook, and it shows 3 stages to the movement of transmission of a signal down an axon. So the action potential starts here, moves here, continues down. Action action potential is moving from left to right. Because of the change that has occurred in the ions, the signal can only move in one direction. Talk about there, I want to give you more information on it tomorrow. For now, just recognize there's myelinated and non-myelinated axons. They function differently. They both pass signals down the axon in one direction. Tomorrow, more information on that. The last one is pretty complicated. You need to know 8 points about synaptic transmission. I'm going to show you a couple of different animations tomorrow about this. For now, let's talk about synaptic transmission. So a synapse is a gap. A synaptic cleft is also another way of stating it. It's a gap between the axon terminal of one nerve cell and the dendrites of another. So they're not directly connected. Think about this. They're passing electrical impulses through your body. So we want to avoid a connection. You're going to establish through the gap signals from one nerve cell to the other, saying pass this signal. And it's going to happen extremely fast. They're not directly touching one another. So there is a synaptic gap that exists between our nerve cells. If we zoom in on that synaptic gap, so here's the axon terminal and the dendrite. Axon terminal and the dendrite of the next cell in line. We have vesicles which contain what's called neurotransmitters. When an action potential moves down the axon and reaches the axon terminal, it stimulates, and it's not in this diagram, stimulates calcium channels, which are membrane proteins that allow calcium to enter the nerve cell. And that calcium stimulates these vesicles to undergo what process where they excrete chemicals? Exocytosis, right. So calcium ions enter, stimulate vesicles to undergo exocytosis, where these neurotransmitters, one we'll talk about tomorrow, is acetylcholine, pass from one nerve cell to the next and initiate the action potential in the next nerve cell. This is simply an introduction. We've got 6 objectives. Tomorrow I'm going to give you some more information about them in order to help us master these through animations. And then you're going to have time to work on the Group 4 project. Questions about these 6 objectives for now? Yeah? Yes. Good question. Can one neuron receive one signal from one other neuron? They can be, actually, there can be synapses between more than one nerve cell, so I shouldn't make it that simple. You can have one nerve cell with dendrites that extend to different nerve cells, but it is only going to transmit one signal at a time. Other questions? All right, tomorrow, 4 animations, practice IB multiple choice questions, and then time for the Group 4 project. Please bring materials with you tomorrow to work on the Group 4 project.